Welcome dear students. Today we will discuss on cell organelles. Right. So first of all, we have all should know what is cell. So cell is a structural and functional unit of life. The human body is made up of trillions of cells and each having a specialized function. As we all know, each cell have different functions, and within the organelles, each organelle having the different functions. So first time it was discovered by Robert Hooke in 1665, and the cell is made up of water, about 2/3 of the cell, and the rest is mixture of molecules like protein, lipid, and carbohydrate. As you all know that, and cell converts the food into molecules by using thousands of different metabolic reactions. Okay. So this is only the prokaryotic cells. This is typically bacterial cells. It labels different flagella, endospore, capsule, cell wall, cell membrane. So all these structures are in prokaryotic cells. And this is a plant cell. So having all the organelles like Golgi vesicles, ribosomes, smooth endoplasmic reticulum, large central vacuole, and cytoplasm, mitochondria. So different organelles are there. So one by one we will study in the next slides. So this is all about the animal cells, and as we all know that there is a difference between the cell wall. Cell wall is present in the plant cells. In animal cases, only the cell membrane is present. That is the difference. Otherwise, almost all organelles are present in the animal cells, right? So, there is some differences between prokaryotic cell and eukaryotic cell. So, in eukaryotes, generally larger in size, complex in structure, membrane-bound nucleus, and membrane segregate functions, and more organized DNA fusion, meiosis takes place, sexual reproduction takes place, and so many different variations or differences between prokaryotic and eukaryotic cells, right? So organelles, these are this is a structure for one organelle. So this is first the cell membrane. So what is cell membrane? It is a lipid bilayer with a polar head and a hydrophobic interior. It means is see, these are the some structure. This is a polar head and these are the tails, right? So this is a lipid bilayer structure. So it's a phospholipid bilayer, it is otherwise called. So these are the layers of different hydrophobic polar heads, and here also hydrophilic polar heads. So these are the different structures, head and tail. These are the tails. So this is all about your cell membrane, how it looks like, right? So it is a head and a tail. One is positive charge, one is negative charge. So this is all about your cell membrane. And see, this is the structure, how the plasma membrane here attached to the protein channels, a protein called channel protein or transport protein, as you can say. And many sugar group like carbohydrates are attached to the protein and liquid. So this is your protein molecules, and this is your liquid. See, these are the liquid layers. So these are the protein molecules. So like that, all the proteins are attached to the plasma membrane itself. So in the cell membrane, you see these are the some structures. It look like in the this is nucleus. As you all know that nucleus is a double membrane eukaryotic cell organelles that contains the genetic material. So the cell chromosome are also confined with it. DNA is present in the chromosomes and it provides genetic information required to the creation of different cell components in addition to the reproduction of life. So it is a important organ, one of the very most important organelles of cells, right? In both the cases, in the plant cases as well as animal eukaryotes or prokaryotes, both the cases it is very important. So this is somehow the nucleus structure. So these are the chromatids. This is centromere, exactly when the chromosome seen under the microscope. So DNA is binding to the histone protein in that cell. So and this is the nuclei divide. You can see this place, the nucleus is divided and followed by the rest of the cell. And bacteria also divide here. This is a fission process. You can see these two, the bacteria divide itself. You can clearly see in these pictures that bacteria divide into both the parts, and this is undergone the fission reactions. And this is cytoplasm and cytoskeleton material outside the nucleus, inside the cell membranes. You can see from the pictures. And cytoskeleton is also provide a rigidity structure, the bones of a cell, so it is also a very important organ. So in cytoskeleton you can see there is the major fibers, microfilaments, like it has some smallest fiber 5 to 6 nanometer wide, fiber of polarity is having, and intermediate filaments this person. So middle side 10 nanometer wide, mostly stable and change less frequently. And this is, you can see the cytoskeleton structure, antiparallelly tetramer, then protofilaments. So this is the structure of cytoskeleton. And you can also see this is a microscopic structure of the microtubule as largest fiber containing 2 to 5 nanometer wide and made up of 2 protein subunits. Okay. And this is all about the cytoskeleton. It contains the microtubules like the spindle fiber and separate the chromosomes as well as flagella that allow the motility, because as you all know that in bacteria cases especially the flagella is only a root that it help in the movement of the bacteria, or it can swim in the water or any medium, right? This is all over central jump. You can see this is a ring-like structure. This is a central jump. That's a microscopic view, and this is called ribosomes. And ribosomes, as you all know that it is both in prokaryotes and eukaryotes. It is called a protein factory of the cell. It is relatively small organelles. So it is of two types. One is 70S, one is 80S. In 70S, in prokaryotes, it also divides into two subunits. One is 30S, 50S, and in eukaryotes, this is your 40S and 60S. So this is sedimentation coefficient. It means when we go for a centrifugation process, it is otherwise called a sedimentation coefficient. It separates the substances by density. Those substances having the more density or heavy, that will separate it and it will settle down in the bottom of the tube. Okay, that's why it is measured by the Vedbar, the scientist Vedbar. That's why it's S is a subunit, which is name according to the scientist. He is given the S is subunit. That is how it is a particle moves to the bottom to the centrifuge. That's why the Vedbar unit is 70 S. So S stands for Vedbar, right? So in this case, one of the important endoplasmic reticulum. So as you all know that there are two types of endoplasmic reticulum, that is called SER or RER, rough endoplasmic reticulum, and smooth endoplasmic reticulum. It is associated with mainly in the carbohydrate metabolism and many other metabolic processes. Also detoxify synthesis of lipids, cholesterol and steroid. And otherwise in drop endoplasmic reticulum, it helps in the flattened sacs, part of the continuous membrane within the cytoplasm of the eukaryotic cell. It plays a central role in the synthesis of protein, and its appearance at the outside surface will be studied with a protein synthesis particle known as biofilm. So this is a Golgi complex. To see, these are the structures of Golgi complexes. So Golgi body is known as a Golgi apparatus. It is a cell organelles that help process and package protein in lipid molecules. So it is also one of the important organelles. So these are the different organelles which help in the each in a specific reactions, or it has a specific function towards the cell. So this is all about your vacuole. Vacuole has a broad definition and it includes a variety of membrane-bound sacs. The membrane are composed of phospholipids. Each organism is slightly different in phospholipids. So in the membrane, embodied in the membrane are proteins. It function mainly it functions as a transport molecules across the membrane or keeps its structure. So it helps also in the transport systems. And this is all lysosome. Lysosome are membrane enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers, that is protein, nucleic acid, carbohydrate and lipid. So lysosome function as a digestive system of the cell, serving both to degrade material taken up by outside of the cell. So this is also a very important organelles in the cell, both in the prokaryotes as well as eukaryotes. And peroxisome both generates and degrades the hydrogen peroxide, detoxify the chemicals and converts some substances, breaks down lungs and fatty acids, and also role in the photorespiration and some biochemical reactions also. And first isolated is a biochemical function. And cell wall, it is one of the important structure, or we can say the organelles. So primary cell wall generally contains cellulose and other polysaccharides. Secondary cell wall, if present, is more rigid and contains lignin and higher cellulose content, that is in the plant cells, because cell wall is present in plant cells, right? So plasmodesmata are cytoplasmic links between plant cell passing through the cell wall. So this is very important structure of the plant cell, that it gives it's made up of peptidoglycan instead of cellulose. So these are the very rigid structures and very important also. So these are the extracellular matrix regarding. So secreted by animal cells, not plants. It contains mostly collagen and proteoglycan. It aids to support the recognition events and it helps the muscle cell function and synapse transformation. So in mitochondria, it is all we know that it is a powerhouse of cell and they help ton of energy we take from the food into energy that can use. So, but there is a more to mitochondria than energy production, because it is only a powerhouse of the cell. ATP is generated from there. In every case, it is very important that mitochondria is such organelles. It helps in the it helps in the generation of ATPs like adenosine triphosphate, the energy currency of the cell, otherwise called as. So this is all about your mitochondria. And chloroplast, as you all know that it is present in the plant cells. Chloroplast is the organelles that contain photosynthetic pigment, and some of the bacteria are also present. The chloroplast are found in all green plants in the algae. They are food producers of plants. These are found in mesophyll cells and they contain a high concentration of chlorophyll that traps sunlight, which cell organelles is present in animal cells. So this is all about your comparison. These are the different characteristics. These are the prokaryotes and eukaryotes. So groups are found. So prokaryotes means bacteria and eukaryotes algae, fungi and protozoa, plants, animals, etc. Size of the cell. So accordingly I have written in the two tables. So flagella, calyx, cell wall, and plasmamembrane, cytoplasm, mesosome, ribosome, chromosome, cell division, sexual reproduction, pseudopodia, DNA basis. So these are about the characteristics, and this is about the and the difference between the prokaryotes and eukaryotes in the both the column. So you go through all the points, and if there is any difficulties, you can directly write in my comment section so that I can change or I will give you the answers. Thank you.